DRI OCC analyzer. So first of all, I will welcome all of you and now request Dr. Andreas to start with the presentation, and then maybe after that, Dr. Ralf will take. Thank you.
Thank you very much. I will just give a very short presentation about Photonion, what we're doing, and what is the special thing about our methods to ionize compounds and combine them with mass spectrometry. So it's about soft ionization mass spectrometry. Now it should move. Yes. So Photonion is a German company founded in 2009 in Germany, in the north part of Germany, in Schwerin. And it is a spin-off of Ralf Zimmerman's analytical group. And we produce customized mass spectrometer systems. And I will explain what we're doing and focus later on aerosol related mass spectrometers. So we are developing soft ionization techniques for mass spectrometry, and we do also research. So where is Schwerin? The headquarters in the north of Germany, as you can see here. And we have production sites in Rostock and also in Munich. So it's all over Germany. And what we do is, you can see here, different mass spectrometers for different applications. We combine them with different sampling technologies, with different instrumentation. And some of these instrumentation we will hear later on aerosol, maybe scientific. So what is the special thing about soft ionization mass spectrometry? You know, mass spectrometers are systems where you measure the masses. So they're very good for identifying compounds in mixtures. And what you do is you separate the mixture with gas chromatography. This takes time. So if you want to do fast analysis without GC, you need special ionization technologies. And one of these is soft ionization. And we do this with light. For example, you see here a mass spectrum of a complex mixture, diesel, with a classical electron impact ionization. This means you get a lot of fragments you cannot identify anymore. So this is why you use normally a separation technique in front, can be gas chromatography or others. If you want to identify mixtures, you need to have something without fragments. Soft ionization is the technology we use. In this case, single photon ionization, UV light, and then you see the same sample, but you see molecular information. You see the masses, molecular masses. And there's another technology called REMP, resonance enhanced multiphoton ionization, which is much more selective, but you can only see some compounds, in our case, polycyclic aromatic hydrocarbons. So the really toxic compounds, also in aerosols, we use REMP ionization for the identification of them. So you can see, compare the spectra, you see the mass, the signal intensity, and you can see it's the same sample, different technologies, ionization technologies, and the REMP gives the information of the pHs. So different fingerprints. And how can we do this? With the single photon ionization can be done with UV lamps, UV lamps, but also with lasers. For the REMP, it's only possible with lasers because you need an intermediate state. You need high energies, high intensity of light, because you have the electron in the intermediate state, and then you have to hit the electron again to get it ionized. So what are the unique selling points from our systems? It's a soft ionization, and we do this in vacuums. We do not ionization in pressure, so we don't have chemical ionization. It's a real physical process. And this helps us for online analysis, fast analysis. So we do not measure main compounds in air like nitrogen, oxygen, CO2, because the energy is too high. We do only measure aromatic compounds or organic compounds. And fast process analysis is then possible. We have combined this technology with different, as I mentioned, different sampling technologies, different instrumentation. I will go quickly over it. Ralf will explain in much more detail what you are more interested in. Yeah, but just that you know what we're doing, I will go quickly over it. So, for example, why do we use soft ionization? Because we want to measure in cigarette the toxic compounds in the cigarettes, and this path by path very fast. So this is why we need the soft ionization technologies. We combine our instrument with a smoking machine for different normal cigarettes, e-cigarettes, even marijuana. In some parts of the world, it's legalized, so they want to see how are the different TIHs concentrations changing path by path in this stuff. So how does it look for a normal cigarette? You see one path by path the signals, and then you can see the butadiene, for example, how it changes with time. For the first path, and then at the end, and how the nicotine is released in the cigarette. This is interesting for people developing cigarettes. We have other applications like in coffee roasting, where we combine the system with a coffee roaster, and there the issue is, where do I stop the roasting? If you have it too long, it's burnt. You see the phenolic line. So this is the mass spectrum, the ion of phenol, and you can see if you wait too long, it will not taste so good anymore. So the question is where to stop, and we combine this with pattern recognition technologies so that you can look where is the optimum of your roasting process. We have also combined this system with a system called aerosol liquid interface, which is a system for measuring. It has lung cells and it measures the toxic compounds with biological aspect, and we just do the gas phase to understand what the cells are exposed to. So different instrumentation, different areas, we use soft ionization with that. So this is the main message. We also have very fast gas chromatography, because there is one issue with mass spectrometry. It can measure only the masses, but isobaric compounds having the same mass, you cannot distinguish. Isobaric compounds not possible to distinguish with mass spectrometry. So this is why we have developed a very fast GC you can put in front. It can separate just in some seconds the compounds. For example, here some monoterpene mixtures separated in 13 seconds. So this is also quasi-online, and then you can see how is the composition of the terpenes. So for non-volatiles, we have also combined the system with a Thermal-Optical Analysis system. This is just heating up the sample. You measure the weight loss, and then you measure what the mass spectrometer is giving you essentially information. And now we're coming to the things which are much more interesting for you probably. So we have coupled our system to an ECOC analyzer from aerosol, and Ralf will explain much more what we're doing here. This is just a setup, and during the different steps where you heat it up, we look for the organic fraction of the OC1, OC2, and OC3 fractions, and look what is coming out of the system. This complex mass spectra, Ralf will explain much more about them. And we have also developed a single aerosol mass spectrometer, where from the air we sample into the vacuum single particles. We have an aerodynamic lens. We have lasers for a sizing unit. We can measure the velocity of the aerosols, and then we predict when they are coming into the ion source. And then we hit them with lasers, and we can ionize the single aerosol. For each aerosol, we can measure the elemental and the organic composition. This can be done 30 times per second. Ralf will explain much more about this. So you get a very complex information for each aerosol. Like here, you can see differences between diesel soot and then wood soot, or so-called tar balls. And the red, you see on the right side, these are the PAHs. We have used also this system for other applications, like for drug and explosive screening, looking for explosive or drug materials like fentanyls in airports. It's now tested in one airport for the baggage and transport control. And as you can see from the mass spectra, they are really different mass spectra. So we hope that we can have a very fast system to select dangerous baggages. And with this is a quick introduction to what Photonion is doing. And now Ralf will give a more detailed explanation of the applications regarding aerosols. Thank you very much. Any questions? I'm here in the afternoon. So Ralf, it's your turn.
Okay. Okay. It seems to work. I have to switch on my microphone, I guess. Otherwise, you will not hear. Put away my phone. Not to have your feedback. Yeah, good afternoon. Thank you very much for having us here. And we are very happy after a visit to the IASTA conference to be here and discuss with you some aspects of aerosol science and aerosol measurement. And I have put a presentation together where we have some aspects of the stuff we presented at IASTA are combined also with the instrumentation, which it's loud, I guess. Down with the instrumentation which was presented already by Andreas, because Photonion is a spin-off of my research group in Rostock. And so let's go in medialis. So just for information, oops, a bit slow, for information, you can see here the research group, which is in Rostock and in Munich. In Munich, it's a Helmholtz Center, but my main position is in Rostock, a quite old university in Germany, founded in 1419. And we have two main topics, mainly instrument development, so new measurement techniques, and the application is aerosol and health effects. And I think in India, this is a big topic. And of course, these two fields are working very much, very closely together. So I will explain in three examples what we are doing now in the following. So I have an introduction. I think we have some PhD students here as well. And so I try to have an overview part as well. Then I will talk about a technique. It was mentioned briefly by Andreas, how to use air-liquid interface exposure of lung cells to directly measure the health effects. In the following, we discuss the single particle mass spectrometer as one aspect to really, or one measurement possibility to really identify the components which are most health relevant on a single aerosol basis. And finally, we come to the coupling to the ECOC analyzer, which is produced by aerosol, and we couple the Photonization Mass Spectrometer to this. And this is then the closing of my talk. So let's start with some overview at the beginning. Well, what's happening in the atmosphere? Maybe, of course, it's you know it very well. You see the boundary layer going up, like we yesterday have seen it, yeah, very nicely. And also the aerosol coming from the plain into the mountains. But what we have is an atmospheric reactor. We have the emissions, natural emissions, biogenic emissions, anthropogenic emissions, which have, of course, very many toxicants. And then we have secondary organic aerosols, which actually play a very important role here in India. And the ambient aerosol, of course, is a mixture of both, because some things are freshly emitted, and others are of course aged, and so we get exposed to this. And we all know, and you know it very well, that we have a big problem here with aerosols and health effects. And this was firstly, let's say, came to public knowledge after the London 1952 incident. There was this heavy smog. And after the smog, it turned out that the people were dying in a much higher rate. So you had a much higher death rate for this period. And interestingly, it's the mouse transmitted. Just maybe you see my mouse here? Yeah. You can see it stayed for a longer time. So not only acute effects, but also long-term effects have been observed. And this actually started a huge amount of studies. You all know this, like this very famous study from Dockery, showing that the life expectancy in very polluted areas is just reduced. And well, this can be all summarized very nicely. And if we look to the risk for premature death, of course, we have a lot of things which are lifestyle associated. Mainly what you eat, how much you do exercise. If you are smoking or drinking alcohol, this is of course the most important effect for your life expectancy. But when you took the environmental aspect, then the ambient PM is actually very important, and the household air pollution. So knowing this, we know now that we have to do something about air pollution, and I've seen really a lot of nice research happening here. It's my second time in India, my first conference in India, and it's really a lot of things are happening here. And the 7 million deaths, which we have annually globally, we have always to keep in mind these are more people per year than during the peak of the COVID pandemic died. So it's actually a silent pandemic, like the WHO is calling it. Why is it so? As I think all this we know quite well. We have the very fine particulates, which are coming in the deeper region of the lungs in an area where actually two lines of cells are making up the border between the air and the inner part. So at that is the only part in the body where cells are coming in direct contact with the air and with particulates. And this thin layer, of course, is very vulnerable with respect to toxicants. And we also can see that there's quite a huge fraction of these particles which we are inhaling are coming in the deeper regions. So if we now would like to study this, we can use a simulated lung approach, namely taking a simulated lung surface, expose this to aerosols and try then to find out what is happening. For this, cells are cultured on transfer inserts. And you can have different cells. It would be a monoculture, for example, but it's also possible to have co-cultures, let's say complex multi-structures where you have macrophages, epithelial cells, endothelial cells, and so on. And by this, you are mimicking the situation in the lung, because we as a trumpet now here, or here shown maybe better, So particles are guided over this lung cell surface, and the deposited particles then induce processes in the cells, and this can be measured after the exposure. And there's a lot of possible things you can do, like inflammation tests, oxidative stress, gene toxicity, epithelial damage assays. So you do a lot of biological work. And mainly we do omics analysis, so transcriptome analysis to deeper understand the process. All this happens in this type of automated aerosol exposure stations, where we have several cabinets. And in these small cabinets, we have always the lung cells, which are prepared. And typically, we expose them for 4 hours with aerosols. For example, from in this mobile biolab, we can do this for specific emissions. And then after that, we harvest the cell, and you can see in this video how now the scientists and the students are actually harvesting the cells after they have been exposed, and they harvest and they get the extracted RNA and the proteins for further analysis. And then from this we can understand what's actually happened to the cells during the exposure. So as mentioned before, it's very often nice to have this type of dual cell lines. So in the body we have 2 cell lines, like here we have the epithelial cells, which is the inner membrane of the alveoli, and the endothelial cell, which is the outer level. Also the blood vessel is of this cell type. And so we even can now have the cells on both sides of the membranes as a co-culture. And as mentioned before, up to we use currently mainly 3 different cell types, sometimes 4 or 5 different cell types, to get a good description of the human lung. Okay, and I will now show you one study with this approach. And this is about secondary organic aerosols. And when we look to the total concentration of organic aerosols, we see that the secondary organic aerosol is much more prominent in many places. And when I look here to India, at least to this study, also here we have much more secondary organic aerosol in the air compared to primary organic aerosol. So typical biogenic precursors are alpha pinene, beta pinene, isoprene, or so plant compounds. But of course, we have also anthropogenic compounds. And the question is now, are anthropogenic or biogenic precursors more toxic? To do so, we take a soot generator and then add the precursor, do an oxidation experiment, and then we get these SOA-coated particles. And we can now take, for example, one anthropogenic and one biogenic precursor. So we have beta-pinene and naphthalene. And as you can see, the soot particles, how they are normally emitted from your combustion sources, are then collapse, because the SOA is condensing and the particles get like a shell of organic material. And then we do the exposure, and let me see. Unfortunately, I don't have this. Okay, I just go over something here. I don't go so much into characterization, just direct to the biology. So now you can see here the dilution of the aerosol. This is 1-to-1 diluted of this, let's say, homemade aged aerosol is one source, and it's diluted here up to 1-to-30. And first we measure here the metabolic activity, and you can see now that our anthropogenic precursor, the naphthalene, which is always here in purple, is much more toxic because the cell viability is really very much reduced. So the same amount of anthropogenic SOA, it makes a stronger effect than the biogenic aerosol. And we see the very same we see now for live dead staining. So this shows us on the cell surface how many dead cells we have. And you can see for naphthalene, we have a much higher concentration of dead cells. So there's more orange fluorescence here by this marker, showing us that the cells are much more impacted by this anthropogenic aerosol. And the same we see for inflammation. So again, we have much higher inflammation measured via interleukin 8, a typical marker for inflammatory processes, compared to the respective biogenic precursor. So again, we have the anthropogenic precursor is worse, and finally also the gene toxicity has the same effect. So what we learned from this is that the SOA is differently toxic depending if it's anthropogenically generated, the precursors, or biogenically generated. And we see even here a difference for co-cultures, because for the co-cultures we've actually seen. So this the co-culture is now the case where we have the two cell lines together. And for this, we see that even the inner cell line, so the endothelium, which is not directly in contact with the air, that we induce genotoxicity here as well. And this means that the toxicity, if you speak it simply, is going into the body already in this model system. So this also is then investigated by different omics analysis. I will not go into detail here. Just one point we see here that the oxidative stress response actually is much higher for this naphthalene SOA, and we see also here some typical markers like the cytochrome 1A1, which is for aromatic compounds which are more expressed, and this shows us that this aerosol has actually more activity. When we now look at a real source, so a real source would be a car engine. Yeah? If you have a car, then you have not only one component, you have a mix of many components. And we do the same experiment. Now we have a car, but the car has, it's a Euro 6 standard, so it has no primary particles due to the particle filter. And what happens then is that we lost our signal here. Okay, guys, I need technical help.
Connect to Wi-Fi once again.
Let's go again.
Maybe just wait for 1 minute, I think. That will come.
Okay, it's connected, yeah?
No, Wi-Fi will be connected. Just connecting Zoom.
Zoom. Again, this one? No, I need all the numbers. Yeah, sorry.
Net,
There he is.
No, it's Wi-Fi is okay. I need the Zoom data.
Okay.
Somebody has to help me with the Zoom login.
Yeah, it's coming to me.
Meeting, yeah. Okay. Okay, I can try nevertheless to continue. What is coming out in this, in the next slide I will show you, is that actually this fluke or this filtration technique is reducing it very much. So it's not toxic anymore as a primary aerosol, but the secondary organic aerosol coming from these clean cars is actually bringing new particles, secondary organic aerosol, and also new tox. And this I will show just in a minute. Maybe we should write down this, that we can log in always again.
Yeah, actually, that how you are getting that information, your lung exposure or that information from this instrument. You have shown the lung exposure or that.
Yeah, so we do it. So we have the lung cells exposed. We harvest the cells, and then it's classical. Let's say, okay. No, it's not. It's wrong. Wrong number. So you have to go. So maybe you can send me the link via email, and I can always click on it. Otherwise, you have to put. No, this is. Yeah, but you have to type in the ID and the numbers.
And I was just wondering on the when you talk in terms of aerosol, and you say, but basically we say the lungs are getting damaged.
Yeah.
So basically they are oxidizing the lungs. What is the actual process is going on?
Well, the process is actually that you have oxidative stress in the beginning.
Yeah.
And then this is more or less the body reacts on this with inflammatory processes.
Yeah.
And this inflammatory process then has a whole cascade of let's say mechanism which follows. So it's like an inflammation without having a pathogen. And this causes in the end that this molecular system actually is going to generate own loss. So you see that this cell, biological molecules are oxidized, so are less functional. And at some point, when you have the DNA damage, which is also oxidative, then if it's too much damage in the cell cannot repair anymore and dies.
So what I'm trying to understand then, so it is mainly the chemical coating on the particle. It is not the physical form. It is the basically chemical coating. So depending on that property,
Yes.
it's chemical is coated.
So the chemistry plays an important role.
That's why.
So of course, it's always the physics as well, because we always have the core of the particles. Without computer audio planning. Okay. So I think I finalize this aspect and then we have the summary.
Okay.
So is now everything okay with the sound as well?
Yeah.
Okay, good. It's again saying that the internet connection is weak. Let's hope. Okay, what we can see that the filtration system is actually reducing the primary particles to zero. But in the secondary organic aerosol, we have again a huge amount of particles formed. And we have the secondary organic aerosol now again to our measurement system. And you can see here, this is a car on the test bed, our oxidation reactor, this cell exposure system, and that's a workbench where the biological tests are done. And in the end, we can see, I only have one example here, but you can see that it's a slight toxicity of the fresh aerosol. So the fresh exhaust, the gases, the NOx is toxic. But the aged aerosol, the secondary organic aerosol, is much more toxic. And that's a problem why we still also in Europe have smog periods. Also, we have all these good Euro 6 cars already. What we can learn from this is that I have to minimize my. That the particle filters alone does not help. We have to emit the gases. And I think it's very much the same for India. The secondary or organic contribution is very high. So with this, to summarize this part, it's shown that we have to look at this issue comprehensively, and that the organic chemistry is very decisive. So different precursors are making differently toxic aerosols. It's not the question how much particles you have, but it depends on the composition of the particles. As mentioned before, Euro 6 cars with PM filter alone doesn't help. And we are currently also in a big European Union project look at different type of emissions to clarify how bad they are really in the real atmosphere. So important is the aging. The atmospheric processes are mattering for the toxicity. The primary PM emission often is more toxic still. So we have the PAHs, the metals, and so on, but it's modulated by the secondary organic components. And as mentioned already, the chemistry makes a big impact on that, what's happening. So we have to look at chemistry. So, and this is now the next part. So in my group, we try to develop different techniques to bring the chemistry better into the measurement processes. And the first approach I go in is this aerosol mass spectrometer. And the idea behind this, there's another dimension, which is very difficult for our understanding on the one hand, but also for the health. That is that the particles are all different. So we have, like you can see here, if you sample particles, you have soot particles, you have inorganic particles, you have dust particles, you have particles which are mixed, you have secondary organic and inorganic components. So the question is now, if a toxicant, the toxic component, is either very concentrated in one particle or is it distributed over all particles? If you take a filter value of the two aerosols, you get the same value. But it makes a big difference. Saying that the connection is breaking down. Okay, I try. It's yeah. Guys. Yeah.
With the line cable.
Okay, now we are back in, I guess, huh? So then I do it again. Okay. Good. Okay. Now we have it. Yeah. Perfect. Good. So what you see is now an important difference is how the pollutants are actually distributed in the particles. And this can be seen very nicely. No, it's not. Something different here. Okay, now it works. Yeah, so this can be seen in this figure. So it makes it different when all the particles only have a little amount of toxicants. The dose for a single cell is not sufficiently high to kill it, simply spoken. If you have these very toxic particles, then it happens that that single cells are actually dying directly from a single particle toxicant, and this causes a very strong response of the whole body, because inflammation is more or less induced. So it's also very important to understand how the pollutants are distributed on the particles. And this can be analyzed by mass spectrometry, namely by a single particle mass spectrometer. And this works like this. The aerosol is put into the mass spectrometer via an inlet directly from the air. So we could place it, for example, here on your station, and would suck the air in. The particles would then be measured by light scattering from two laser beams. So the particles are flying in here, and the time difference between the two scattering signals gives us its velocity, the size, and also the time when the particle is in the center of the ion source. And then A laser pulse is actually ionizing this particle, and you get negative and positive ions from that single particle, which is giving you the information on the composition. So what you get is like you get information on metals like iron, potassium, sodium. This is now a single particle, or on nitrate, sulfate, or soot carbon clusters. You see C2, C3, C4. These are signals from soot. Or here you can see potassium, yeah, and so also some organic fragments, nitrate, sulfate, and so on. And it's actually very nice because you have, well, after a day of measurements, you have 100,000 of single particle information, which you can then, via cluster analysis software, you can sort and make a source apportionment of your air masses. There are some limitations. We are not seeing the most important compounds for health, the PIHs. We don't see with this approach. So organic compounds are disintegrated in this process. But of course, very nice source apportionment can be done. This is one example here from Rostock. We have the Baltic Sea, and you see here wind coming and ships are coming by. And when the ships are using heavy fuel oil, where vanadium and iron is present, we can see that then here the iron and vanadium containing particles are peaking. So we can see from downtown Rostock when the ship is going by, and we see the signature in the single particle mass spectra. So we see that here, I don't know, 50 or 100 particles is the signature, and we know we can back calculate which ship actually was responsible for this emission. It also can be done in a research cruise. We looked with a research vessel from Germany. We looked at some shipping routes and tried to identify if specific by-coming ships are using, for example, heavy fuel oil or not. You have to know that heavy fuel oil is not allowed anymore. It's the Baltic Sea, so we have a heavy fuel oil ban. And we look then if those ships are using scrubber cleaning or not. So if they have legal operation, yes or no. But of course, our main interest is health effects. And as I mentioned before, what is really missing is the organic coating information, or the PIHs. Because this is one of the driver of the toxicity, specifically of the long-term effects like lung cancer. And the question is if we can add the information into this technology. And this was realized actually in my research group by having such a dual-step ionization, where the particles are first heated up on the fly. They're going into the ion source, heated up by infrared laser. Then we have the normal process, but the ionization is done with one laser. We first ionize this plume of PIHs, which are dissolved, and then it's refocused on the particle core, where plasma ionization shows us the metals and the salts. So what we get are mass spectra like this. Is a single particle of 400 nanometers, and you can see the signature of PIHs now on single particles. This was a slide shown before, just showing that different sources have different PIH profiles as well. Very interesting is for ship emission that you can see, for example, when we look at many different particles from ship engine, that the pattern of this PIHs always stay constant. So the pattern of the PIHs is good to identify the source, while the inorganic part varies. We have soot particles, or we have phosphate-rich particles from lubrication oil, or we have nitrate-rich particles from the NOx formation, but the PIH pattern is always the same. And the same holds same pattern for this single source, and this needed to be tested just to make sure that we can use it as a source apportionment tool. And indeed it works, and it's just now one example how it looks. You can see over some hours, you can see every gray dot now is one particle measured with a pH signature. And every red dot now is a particle with a signature of ship emission. And you can see we have a broad ship emission background here. This is a background station in Sweden, actually. That's a seaside. And here we see a ferry going by, and we can see the signature of a ship plume, which is then shown by the signature we have seen before from this phenon trains, which are coming from the ship emissions. For health effect, quite important is that at this remote site here in Sweden, we see a huge amount of particles. We have measured 1.3 million particles in 10 days. Also, the air was very clean there, very, very clean. Yeah? So at the same time, we would have measured probably 100 million particles in India, because this is really, the air is maybe, total is definitely below 5 micrograms. This is below the World Organization standard, very healthy environment. But we see anyway that the particle cores, this is a fraction, we have sea salt, we have different, let's say, organics, we have iron, nitrate, and so on. These are the particle cores, the inorganic part. And the outer ring shows the fraction of those particles which are carrying pHs enlarged by a factor of 10. This means that only 1% or 2% of those particles actually have pHs, most of them not. And so we still have a high concentration of the toxicants in some particles. And this could be different in cities where we have maybe 60% of particles which have pHs. Of course, we can then use here again, we have this the ship emission particles. This for source apportionment. We have biomass burning or industry, high temperature pH patterns, which is coming from coal burning in facilities, in big plants. And if you have smoldering biomass burning or trash burning, you have this type of smoldering pattern. And this allows to address this very nicely. In summary, this approach allows to measure the particles on a single particle basis. You measure soot, pHs, and metals, which are the most toxic components on the particles, and it's the most relevant particle size also for PM toxicant lung deposition. So it's a bit focused on the health relevance. And you can address a mixing state, so you can understand if your pollutant is concentrated on a small fraction or if it's totally, let's say, broadly distributed. And then you can estimate how strong the cellular impact would be of this. And as mentioned by Andreas, so also now new applications are like aerosol detection or hazardous substances measurement. And finally, I will come to a technique for filter measurement, and this is a Thermal-Optical Carbon Analyzer for elemental carbon and organic carbon, coupled to a Photoionization Mass Spectrometer, so that we can also measure the chemical profile in the same time when we measure these filters. So the idea is that we use photoionization, and this has been explained nicely by Andreas before. So there are two approaches, either a single photon approach or our resonance enhanced multiphoton approach, which is showing the aromatic molecules. And this VOV light approach gives an overview over the organic compounds, but less specific. And this all happens in the ion source. We have a gas inlet, and the light is directed either by laser or lamp into this ion source. The ions are then accelerated, and we do an time of flight mass spectrometry measurement of that. So we can see fingerprints, like when you see coffee roasting or biomass pyrolysis or cigarette smoke. you can get a fingerprint, and you can then do time-resolved measurements, so you see how compounds, how gaseous compounds are actually evolving. So one example here is like we had the ship emissions before. You can see here this are the gaseous PIHs in ship emission exhaust gas. And when the ship emission ship engine is operated with clean diesel fuel, classical normal diesel fuel, you can see that the emission is relatively low. When they switch to heavy fuel oil, we have a huge increase of the emission of different PIHs. You can see this is a penetrating pattern we actually later on have seen in our single particle mass spectrometry results. So, but what can we do now with the technology in combination with the EC/OC analysis? So this DRI EC/OC analyzer is working in a thermal protocol where it is classical EC/OC protocol. We put a filter in an oven. We have then in the end a total carbon measurement as a fraction of temperature steps. This is one of the historical work for Zhubi Chao, which actually introduced this technology she promoted it very largely. And you can see we have now OC1, 2, 3, 4, and also elemental carbon on it. But of course, we don't know what it is. We only know the amount of carbon. So the question is now, can we do something more on this and best online during the measurement itself? And again, we have seen that we would like to know something about the elemental carbon for health reasons, because the soot is one of the drivers which is used in EPI. Then the organic carbon, we have seen before how the organic carbon is contributing to health effects. And of course, one specific compound class of interest are the PIHs. And question is how can we put this together? When we now take the OC EC analyzer, which is here shown as a schematic and here as an instrument view, this can be coupled to the mass spectrometer, but we have just right before we have the evolvement of the gases, we take a small fraction of the gas in the ion source, and then we do photoionization. And the photoionization gives us molecular fingerprints. And now we can, during this EC/OC process, we can measure the chemical information. You can see how it looks like. The systems are hyphenated then. It's a mass spectrometer unit. And this is a thermal-optical analyzer. And we get now a complex, let's say, information, which can be, however, easily understood and also used. This is a classical EC/OC information. You have here the heating temperature protocol, and you have then the evolved organic carbon. When you now look on the mass spectra in a 2D plot, this means that we have here the mass numbers, and here we have the time. We can see that there's a specific fingerprint of these organic compounds which are involved coming from this filter. We now would like to better understand it, or more easy to read. We just we sum the mass spectra of the OC1, the OC2, and OC3. And this is now done with the ion source, which specifically looks at the PIHs. And we have seen the PIHs have two important features. They are good for source apportionment, and they are good for health effect prediction. And what we see in OC1, we see mainly desorption. So we see the thermal desorption of, let's say, medium heavy PIHs. In OC2, we see desorption of the heavier PIHs, but we see also smaller molecules which are actually pyrolysis of the oligomers. And finally, we get more pyrolysis in OC3 and even higher PIHs. So we also can see if these PIHs actually are there, just dissolved, or if they are formed by the thermal process. And this best can be seen if we now look at the different molecular masses. And here we can see, for example, for chrysin mass, it's very much clear it's dissolved. And later in the pyrolysis mode, there's quite little. On the other hand, phenanthrin is mainly formed in the pyrolysis part, so it's not these phenanthrin units are actually embedded in larger structures, not bioavailable, not toxic. Or here for phenol, again, you can see it's mainly a pyrolysis product. So this gives you a very nice overview of the chemistry. And of course, it can be used just to compare different fuels. You get fully different fingerprints for different fuel emissions, so ship emissions from different fuels. You can see that the fuels are mainly presented by the unburned hydrocarbons, which are always slipping during the combustion process. Or you can look at wood combustion. Here, different wood types give different fingerprints. And then you can do a cluster analysis, can tell you which type of wood actually is coming from these fingerprints. Finally, what additionally can be done during this measurement of the organic tracers is that we are doing electron impact ionization, like in the AMS mass spectrometer from Aerodyne. And here we are then looking mainly at mass number 43, which gives us a C2H3O plus and the C3H7 plus. But it's separated at about mass resolution 1800. You can separate this. And this is an important aspect if we have oxidized aerosol or hydrocarbon aerosol. And this gives you more or less information how strong the oxidation of your aerosol is, like in the AMS. And this is given as a factor as well. We can see, for example, if you have a chamber experiment with oxidation, you can see that the oxidation peak is relatively increased with respect to fresh wood combustion here. And for aged, you can see it's just higher. If you integrate it, you can calculate the O to C ratio from that data. So finally, to summarize this, the definition of this DRI TOCA analyzer, this Photonic Mass Spectrometry was established actually together with the colleagues in Reno and Judy Chow and co-workers and in Germany with my group. And it's a novel system in that way that we get also the chemical information during the ECOC measurement. It adds the chemical information. We can use parallel different ionization methods so that we can, for example, do the impact ionization for oxidation state and the aromatics for the PIH fingerprints and the source apportionment. It's good for PM source apportionment. And it addresses directly toxicity because we have the PIHs, the soot, and also the organic aerosols covered. And in this, it's also now commercially available. Now it's from my research group, it's in the hand of Andreas and also of the colleagues from Aerosol to integrate it and make use of it for aerosol research. So in total, the lecture I would like to summarize novel concepts. We think, or our aim is always should keep the health effects into the focus, because that's the most urgent problem with aerosol emissions. So with air-liquid interface, online cell exposure, we have a new approach to directly assess the toxicity of emissions. And we have learned from this that the chemistry is very important. We learned that surface oxidation and organic compounds are important. And in this respect, I think it's good to evaluate the different sources and also to evaluate different abatement technologies. We can use a lot of money to make something clean, but when then the secondary aerosol is nevertheless more toxic, you should direct your research and abatement technology on the right target. The single particle mass spectrometry allows now this measurement for the first time of internal-external mixture, and also addresses now the most important toxicants, soot metals and PAHs. And the coupling of the thermal-optical carbon analyzer with photoionization mass spectrometry is a nice addition to this TOCA approach, as you have at the same time the chemical fingerprints and source information you normally would only get with very, let's say, time-consuming and expensive offline measurements. With this, I would like to thank you for the audience. I know it was a broad topic from biology to chemistry. This is just a working group in Rostock and Munich together. And this was before COVID. Actually, last time we have been together for a big group meeting, and we have to redo it in the near future. And actually, if somebody is interested, we are currently in the next time have quite some PhD and postdoc positions open at Rostock in the fields I just reported. And of course, finally, I would say also some word to Photonion, because this company is run, except of the CEO, who has an own company and is not actually my former PhD student. We are thinking of the same age about. Yeah, but most of the people doing the work are former PhD students now, which are developing those instruments for aerosol research. And with this, I thank you for the audience. And yes, I'm open for discussion and questions as well.
Thank you. Thank you, Dr. Rao. Now I will request Ms. Absa to give some brief information about DRI multi-wavelength OCC analyzer, please. Thank you.
I just a very quick query. Is it possible to identify whether the sample you are taking is internally mixed or externally mixed?
For the sample for the TOCA-PIMS, or
I mean any sample you collect.
Yeah.
So is it
So for the collection, it's actually nearly it's not possible. So it's only possible with the online mass spectrometer. For the collection, there's only one possibility for some components. You can collect on substrate and then look with electron microscopy, and then you can do an EDX of each particle.
Yeah, and then make this separate analysis.
This is enormously difficult, because once you have mixed them on the filter, they are mixed.
Also they are mixed.
They're mixed. So you have to really measure them on the fly, particle by particle. It's the only possible method. Well, unfortunately or fortunately, so but it's of course it's very interesting to understand. It's a new dimension in aerosol research, for sure. Yes. Okay. So, okay, I'm actually also, I have a small web meeting now with some colleagues. I'm perfect in time for this, but I will be able for other questions later on. I'll just stay outside, how it will work from here. I am sure. To better connect them to the system, because the internet was really sometimes, but you have to stop sharing.
Yes. That's okay. The pyramid? Oh, it's you. Microphone. Okay. So good afternoon to everyone.
Mic is not on. Mic.
Yeah, it's on. Should I put it closer?
Yeah, you put it a little up. Maybe it's.
Okay. Okay. Good afternoon. So I'm Asta Gregoric from Aerosol company. And I'm going to present a little bit more specific our instrumentation. Since we are focused, our company is focused on carbonaceous aerosol measurements, not only measurements, but development of instrumentation for carbonaceous aerosol measurements. Okay. So Aerosol is a small company located in Slovenia. It is the owner is Tony Hansen from US, and he's also the inventor of etalometers, which was our main field until we got the privilege for working and developing the DRI instruments. So in total, we are now about 35 employees, and we also have a very big, I mean, for this small company, quite big research and development group, and with 6 people, actually not 7, as it's written here, but 6 people holding also the PhD. We also publish a lot of things, a lot of papers. We are involved in several collaborations within Europe and also worldwide. So this is what we do. So the outline of my talk will be first a little bit more introduction into carbonaceous aerosols, but Professor Zimmerman already explained quite a lot about the whole topic of carbonaceous aerosols, so I will be quite brief here. Then I will present the techniques for measurement of organic and elemental carbon, and then I will switch to more specific technical topic on the multi-wavelength thermal-optical OCEC analyzer DRI. Okay. So the carbonaceous aerosols are very diverse. They impact public health, air quality, visibility, and so on. So, and they are very important climate forcing. The main separation is to the elemental and organic aerosols. So the organic carbon and elemental carbon together are forming the total carbon. So from the point of view of measurement techniques, we have 2 approaches. One is, I'm sorry, 2 approaches. One possibility is to, if we are measuring the optical properties, we can separate carbonaceous aerosols to black carbon, which is the most absorbing, and it absorbs evenly through the whole spectrum. Then we have the organic aerosols, which part of them are absorbing, and they absorb in the short wavelength and ultraviolet part, and this is so-called brown carbon. Brown carbon, and then we have part of the organic aerosols which are not absorbing, and we cannot measure them by optical techniques. On the other hand, we can measure the refractory properties of aerosols, and we have, from this point of view, we have the elemental carbon, which is actually similar as black carbon, just measured with different techniques. And then we have the organic carbon, which is less refractory. And based on this, we have the thermal-optical technique for measurements for the separation of organic carbon and elemental carbon. And here I'm presenting, from here on, I will talk about the DRI 2015 Series 2 model. But of course, This technique, what I'm presenting here is more general. So in very general terms, what we are doing is to put in the sample, filter sample into the oven. And then we use two techniques. First, we use the optical technique, so the laser source, and we can detect the transmittance and the reflectance from the filter. Meanwhile, we are increasing the temperature, so the organic aerosols are evaporated from the filter in different steps. When during the evaporation, the evaporated sample travels to the manganese oxide, which oxidizes the sample to the CO2, which is then measured by the CO2 sensor. And in this case we have the non-dissipative infrared sensor to detect the CO2 pulse. Okay, so this DRI system was first developed by the Desert Research Institute, and from here also the name come. This is here you can see the 2001 model, and in the 2015 model was awarded to aerosol for manufacturing. From this point here until 2020, we improved the system with several modifications. So we have now the DRI 2015 Series 2 model, and I will explain later what are these improvements and modifications. And in 2023, so this year, we developed also the autoloader. This is this additional part here, which allows you to have several samples with auto-loading system. So with this, you can continuously analyze several samples. So the principle of operation, so the main features, the model 2015 measures the reflected and transmitted signal at 7 wavelengths from 405 to 980 nanometers. And this additional information, optical information, can be also used for quantification of brown carbon in each sample. So we have more information, not only the organic carbon and the elemental carbon, but we also have the optical properties of the brown carbon. It uses the helium gas for the analysis. We'll explain later on how it works. And the CO2 is detected by the sensor. Here you can see also the detection limits and the measurement range is from 0.1 to 1,000 micrograms per square centimeters. So during the what happens during the increase of temperature, what we do is, of course, increasing the temperature with different steps according to different thermal protocols. And during each step, the organic aerosols are evaporated and then measured as CO2 gas. First part of the analysis is done in the helium atmosphere, so without oxygen, in order to prevent the oxidation. So in this case, we are measuring organic carbon, as already Professor Zimmerman explained before. Part of the organics is transformed into the pyrolytic carbon. That's why the optical signal, which you see here, is first decreasing, and then after that, when the oxygen is added, elemental carbon is burned, and we get the increase of transmittance. So the filter gets wide. Here is a little bit more better explanation actually. So we have here the starting sample. During the OC evaporization, during different OC phases, the temperature increases, the transmittance signal decreases due to the formation of pyrolytic carbon. Then we add oxygen, And first, the pyritic carbon is measured, which is part of the organic carbon. So this pyritic carbon is added to the organic carbon fraction measured before, and after that, the elemental carbon combustion happens. So this OC-EC split is very important for determination of the split between organic carbon and elemental carbon. And that's why we are using the transmittance signal, and the split point is at the point where transmittance signal gets to the same level as was at the beginning of the before the sample analysis started. So here are different thermal protocols that are used. So the most important are NIOSH, Improve, and user protocol. There are some differences between them, and also the OC-EC split is a little bit depends on the thermal protocol that is used. And there are several publications on the comparison of the protocols and so on. During the analysis, this is what the thermogram looks like. So we have here the temperature increase, the CO2 pulse with blue. And in yellow, we see the transmittance signal at 635, which is usually used for the OC-EC split. You also get instant results at the end of the sample. And the OC-EC analysis is done for different split points according to different optical signals. So you have all the results together for the analysis. So based on the transmittance signal, you can also determine the brown carbon concentration, or at least the brown carbon absorption. And this is done in the way that here on the left, you see the transmittance signal. It starts at a specific point. So it depends, of course, on the instrument and the sample. Then the signal decreases due to the formation of pyritic carbon, and then increases again. If we want to determine the attenuation coefficient on the fresh filter, or the fresh sample on the filter, we have to use the information about the intensity of light at the beginning and at the end. So the end part means when the filter is already white. So this is the reference signal, and the ratio between the reference signal and the intensity at the beginning is the attenuation. If we have the information, since we have the information about the area of the filter which we used, and the volume of the sample that was sampled, we can calculate the attenuation coefficient. From the attenuation coefficient, so there is a little bit difference between attenuation coefficient and absorption coefficient, which is caused by the filter, and this is this is also published by Judy Chow. From here, so what we get is the attenuation and attenuation coefficient, which can be then used for the brown carbon analysis. So here you see one specific sample and the attenuation coefficient here. Oh, sorry, this is the absorption coefficient. So the difference of the absorption coefficient measured at different wavelengths, and according to the brown carbon model, you can separate the absorption by black carbon and absorption by brown carbon. The model that is used with the DRI software is this one that is presented here. So here are three different unknown parameters. So parameters that are tuned by the model. This is angstrom exponent by black carbon, angstrom exponent by brown carbon, and the intensity of these two. Armstrong exponent of black carbon is fixed to 1. So we have 3 unknown parameters: Armstrong exponent of brown carbon, intensities, and the based on this the model, we can separate the brown carbon and black carbon absorption at different wavelengths. And since we also have the mass of organic carbon measured, we can determine also the MAC values, or mass absorption cross section. Here are different cases which are also published by Judy Chow. And what we get from this is mass absorption cross section of brown carbon, the Armstrong exponent of brown carbon, and the absorption fraction of brown carbon for each wavelength. So starting from the old version of the DRI 2001 to DRI 2015 series 2, what changed is the sample and oxygen oven design. Then we have optimized heating models. We are eliminating the cold spots between both ovens. Then isolation is improved, and also the electrical connectors are improved. So these are all the improvements on the hardware side. There is also efficient cooling system, which is very important for shorter analysis preparation. So we can shift from one analysis to the other, to the next one in a shorter time. And there's also possibility for high sensitivity mode by changing the sample area size. So this sample board design is in the 2015 series 2 model. There is a new CO2 sensor used. So this is LIFER, which is more sensitive, and it allows for more precise measurements. And it also allows for faster analysis preparation, since it doesn't need the zero. Okay, so these are all the hardware improvements. And of course, the DRI 2015 new series, so series 2, was tested and compared to the old version with several samples, where we show that the comparison is very good. We have the slope close to 1 for all the samples. So highly loaded samples, samples with less loading, and so. And the last improvement is the autoloader. So the autoloader is this part that you see here. So this is the sample holder. What you do is to take this sample holder, you make punches of 50 filters, you put it in this numbered sheet here. So each hole is numbered. You prepare a database of your filters in the special database, and then you run the analysis. What is also important is that we added the cooling, so the sample holder keeps the temperature below 23 degrees Celsius, and this compartment is in overpressure to decrease contamination from the room. And this system then also allows that you put inside these filters. You also can add blank filters in. The design you can see here. So this is this sample holder, sample cassette. Below that there are fans, and here is an arm which shifts the sample into the sample board, and then that's what. And that's it from my side. So here is just the last slide, which is the information about coupling the DRI to the mass spectrometry. So what we do is actually this, what is being seen here is the quartz with the sample, and before the sample goes to the manganese oxide for the oxidation, we take a small amount of the sample and drive this sample to the mass spectrometry. And the second part is already covered by my colleagues from Photonion at Rostock University. So thank you for your attention. And if you have any questions.
Any questions? Okay, thank you very much for your kind attention, for actually listening patiently, 3 back-to-back presentations. Thank you very much. Yeah. Or any question you can write to Dr. Ralf or Andreas or also us if you have doubts. Thank you. Thank you, sir, for thank you very much.
How you will get the information about water soluble organic carbons? Is it possible to get with this?
I don't think so. So for the water soluble